Bismillahirrahmanirrahim. Assalamu alaikum. The topic that we're going to discuss for the practicals today is bench test. So these are actually a number of tests that we perform to differentiate between different bacteria and to identify the specific species of the bacteria, which includes a catalase test, coagulase test, oxidase test and bile solubility test. So first of all, we're going to discuss about the catalase test. This is a test which is used to differentiate those bacteria that produce the enzyme catalase such as the staphylococcus from non-catalase producing bacteria such as the streptococci. So we know that the gram-positive cocci are staphylococcus and the streptococcus. So we are going to differentiate them using this test called catalase test, because the staph species are catalase positive as they produce the catalase enzyme, while the strep species and enterococci are catalase negative as they don't produce catalase enzyme. What is the principle of this test? Catalase is an enzyme that degrades hydrogen peroxide with the release of water and oxygen. So when oxygen is released, bubbling is formed, which indicates that the test is positive. Now we have two methods, the slide method and the tube method. In slide method, we need a slide and of course the test or organism. And how are we going to perform it? With an inoculated needle or wooden applicator stick, which should be sterile. We're going to transfer a growth from the center of the colony to the surface of the glass slide, and then we're going to add 1 drop of 3% hydrogen peroxide and observe for bubble formation. Then for the tube method, we are going to pour 1 ml of hydrogen peroxide solution into a test tube, and then we are going to use a sterile wooden stick or glass rod and remove the good growth of test organism and then immerse it into the solution. And then we are going to look for any bubble formation along the rod or the wooden stick. How are we going to interpret the results? If we see the bubbles, that means the test is positive, because I told you that the catalase enzyme is going to perform the reaction which is the conversion of the hydrogen peroxide into water and oxygen, and thus the oxygen formation will form some bubbles, and that shows that the test is positive. And it is positive in case of staph species, while it is negative in case of strep and enterococci. And this shows the slide method, the positive test as shown the bubble formations, and the catalase negative, there is no bubble formation in this. While in tube method, again you can see that the bubble formation shows the catalase positive, while no bubble formation shows catalase negative. We need to take some precautions performing these practicals. The first one is that we have to use a sterile wooden stick or glass rod so that we don't contaminate the test. And then the second one is that we don't pick up the colonies deep from the blood agar, that may give false positive results. The second test is coagulase test. This test is used to differentiate Staph aureus. Now the three of the Staphylococcus species, the Staph aureus, Staph epidermidis and the Staph saprophyticus. The Staph aureus produces coagulase enzyme, so it can be differentiated from the rest of the two that are negative for coagulase enzyme. That means they don't have this enzyme. What is the principle? The coagulase enzyme, which is present in Staph aureus species, converts fibrinogen into fibrin and helps in the formation of fibrin clot. Now there are two types of coagulase that are produced by the strains of Staph aureus: the bound coagulase and the free coagulase. Bound coagulase is detected by the slide method, while the free coagulase is detected by the test tube method. So the bound coagulase converts fibrinogen into fibrin clot without requiring the coagulase reacting factor, and it is bound to the bacterial cell wall and it reacts directly to the fibrinogen. It can be detected by clumping of the bacteria in the rapid slide test, while free coagulase is an extracellular protein enzyme of the bacteria which is not bound to the bacterial cell wall, which converts fibrinogen to fibrin by activating the coagulase reacting factor. In this we didn't require this factor, while in this we required this factor which is present in the plasma. Now free coagulase is detected by the presence of fibrin clot in the test tube method. So how are we going to perform it? For this, of course, first of all we need some slide to perform for the slide test. We're going to place and we're going to label the slide into two portions, T for the test and C for control. On one side we're going to label C and the other side we're going to label T. And then we're going to place a drop of saline on each of the sides of the test slide, and then we're going to emulsify the colony of test organism in each drop to make a smooth suspension. We're going to take a colony and we're going to emulsify on each of these drops, and then add a drop of plasma to suspension T. The one that we label T, we're going to add a drop of plasma, and then we are going to look for the clumping of organisms within 10 seconds. The side that we labeled as C, we are not going to add plasma into it. Results: if we see clumping as shown here, that means test is positive, and if there is no clumping, that means test is negative. Clumping again happens to be because of the formation of fibrin clots because of the help of coagulase enzyme, which converts fibrinogen into fibrin. Now the test tube method, we are going to dilute the plasma in 10 ml of normal saline and we're going to keep it aside. Then we're going to take 3 tubes and label them as T for the test organism, positive for positive control and negative for negative control. And then we are going to pipette out 0.5 ml of diluted plasma in each of the 3 tubes. The first tube is going to have 5 drops of test organism broth. The second tube, which is labeled as positive, we're going to add 5 drops of Staph aureus broth in that. And the third one, which was labeled as negative, we're going to add 5 drops of sterile broth that shows no organism to that labeled as negative. And then we're going to mix all of them and we're going to incubate them at 37 degrees Celsius for about 1 hour. And after 1 hour, we're going to look for any clotting. If we don't see any clotting, then again we're going to incubate it, and after every half hour we're going to examine and look for clotting. This is to be continued for about 6 hours. And if we see a clot formation after all the 6 hours that we have completed, the test is going to be positive. As you can see that here we see the clot formation. This is coagulase positive. That means that the clot formation is positive, and of course the Staph aureus species are considered to be coagulase positive because they form coagulase the clot formation. Now the negative one, in which there is no clot formation, is going to be the negative test. And the other 2 species of the staphylococci, the Staph epidermidis and Staph saprophyticus, are going to be the coagulase negative. Quality control again, because plasma must be treated with known control organism at the time of test, which is going to help us to compare